sperm must travel through this layer of follicle cells before it reaches the zona pellucida, the extracellular matrix of the eggs.
Cleavage is a series of rapid cell division during early development. The cell cycle consists primarily of the DNA synthesis and mitosis phase. G1 and G2 phases are essentially skipped, and little or no protein synthesis occurs. This makes no increment in the mass. Cleavage partitions the cytoplasm of the large fertilized egg into many smaller cells called blastomeres. Each blastomere much smaller than the entire egg. It can make enough RNA to program further development. 5 to 7 cleavage divisions produce a hollow balls of cells called blastula. Blastula surrounding a fluid-filled cavity called blastosome.
After 30 hours of fertilization, it undergoes its first mitotic division to produce 2 identical cells. After 40 hours, it's become 4-cell stage. In the next 2.5 days, it's become 8-cell stage. Then, in the next 3 days, these mitotic divisions continue, eventually forming the 32-cell stage called the morula. The morula will continue dividing until it forms the blastula. For the early blastula, after further cleavage divisions, the embryo is a multicellular ball that is still surrounded by the fertilization envelope. The blastocele has begun to form in the center. For the later blastula, a single layer of cells surround a large blastocele.
After cleavage complete, implantation takes place at the uterus. At this stage, the embryo is called blastocyst. A group of cells called inner cell mass clustered at the end of the blastocyst cavity. The inner cell mass will develop into embryo proper, which are source of embryonic stem cells 9.
Implantation initiated by the trophoblast, outer epithelium of the blastocyst, 7 days after fertilization. Trophoblasts secrete enzymes to break down the molecules of the endometrium lining of the uterus and allowing invasion by the blastocyst. Trophoblasts also extend finger-like projections that cause capillaries in endometrium to spill out blood. Then, trophoblast tissues capture that blood. Inner cell mass of blastocyst forms a flat disc with an inner layer of the cells epiblast and the outer layer hypoblast.
After implantation complete, gastrulation begins. Some epiblast cells remain as ectoderm on the surface. Others move inward through a primitive streak to form mesoderm and endoderm. End of gastrulation, embryonic germ layers have formed, extraembryonic mesoderm, and 4 distinct extraembryonic surround the embryo. Development proceeds. Cells of invading trophoblast, the epiblast, and the adjacent endometrial tissue all become placenta. 4. Exchange of nutrients, gases, and nitrogenous waste between developing embryo and the mother.
ثم جعلناه نطفة في قرار مكين ثم خلقنا النطفة علقة ونقر في الأرحام ما نشاء إلى أجل مسمى يخلقكم في بطون أمهاتكم خلقا من بعد خلق في ظلمات ثلاث